Good morning. Guys, start collecting to hear lecture, to listen to the lecture. Guys, first division, are you there? Guys? Good morning, guys. Good morning. Thank you for coming. So we start our lecture right now. And as you remember, I hope we study right now special virology, and our lecture is about herpesviruses. And on the very first slide of our lecture, you can see the most typical clinical presentation of one of most common herpes virus infection. You can see a batch of vesicles, right? So that then form crust and then resolve within 10 days. And these are the electron microphotograph of the virus that cause this herpes virus type 1 infection. And this is the memo, my dears. So we have 8 herpes viruses, but irrespectively of the type and the classification of the herpes viruses, all of them stay with us forever. And quite often those viruses spread through the sexual intercourse or kissing. So, and as a result, love comes and goes, but the herpes lasts forever. And that is true about all herpes viruses we will discuss today. So, first, this is a really very large group and very important because the morbidity of herpes virus infection around the world is very high. So almost 95% of people have antibodies against HSV1 or a bit less for HSV2, and the rest 6 are also very common. So, and they are placed in one single family because of the very common basic characteristics. So they are very large. This is one of the largest type of the herpes of the viruses. So, and all herpes viruses share the common viral morphology, mode of transcription. All those viruses are ubiquitous. I hope you know what does it mean. It means so we don't have any endemic areas for their multiplication, for their prevalence. So they are very common everywhere in the world. So that's why we call them ubiquitous. They can be found everywhere. So interestingly, that in their life cycle, herpes viruses may cause different types of infections. And the types we studied in the first semester when we studied infection and infectious process. So this is regarding the interaction between the host cell and the virus. So in their interaction with the host cell, herpes viruses may cause variety of different types. For example, that's typically that herpes viruses cause so-called lytic infection. What does it mean? It means when virus multiply in the cell, they are released by cell lysis, and it causes usually some wounds development or some injuries development in the tissues. So then the second type, they can cause persistent infection. What does it mean? they may persist in some particular type of cells, and different types of herpesviruses have or demonstrate tropism for different types of tissues, by the way. So they are not all neurotropic or lymphotropic. So we divide them into neurotropic and lymphotropic. So where they may cause persistence. Then also they may cause latent or recurrent infections. What does it mean? The virus can stay in the body in depressed state without multiplication. Then when some stimuli activate the virus, it starts replicating again and cause so-called recurrent or reactivated infection. So then, apart from this, some of the herpesviruses, for example, Herpesviruses type 4 Epstein-Barr virus and type 8 Kaposi sarcoma associated viruses, they can cause so-called immortalizing infections. What does it mean, immortalizing? It means the particular type of herpes virus may make the cell immortal, or in another words, they make cell to undergo oncogenic transformation. So the normal cell may be converted by the Epstein-Barr virus, for example, into tumor cell. And that is well-known characteristic of Epstein-Barr virus. So altogether, viruses may cause a wide or broad range of infections, from asymptomatic or very mild to the very severe illness. And if we get severe illness, it may cause very strong immunocompromission. And one more thing, that usually some of the herpes virus infections, especially as we said, Epstein-Barr infection, may cause very severe illnesses in immunosuppressed individuals. So because this virus is used to persist in the human body, they depend so much on the state of immunity of the body. One, they cannot be eradicated from the body. So they can be either kept under control, or if they are not kept under the control, so they from time to time may be reactivated and cause recurrent infection in immunosuppressed. And while we will be discussing the diseases caused by these viruses, we will underline always, and you should do it on examination when you answer this question. So you have to demonstrate what kind of diseases are caused in people with normal immunity, immunocompetent individuals, and what kind of immunity is called in immunosuppressed individuals. And that will be quite different. So first words about we cover the basic features. So the virions are spherical usually, up to 200 or 250 nanometers in diameter. That's quite large for the viruses. And they are very complex. So it means that we studied some viruses that may have 5, 7 different types of proteins, including non-structural proteins. But those viruses are really very complex. So if we take a look at the virion, what that is, infectious virus particle, it is usually built up from around 35 different structural proteins. That's quite a lot, by the way, to compare with other viruses. So interestingly, these DNA viruses contain several types of enzymes. And these enzymes are very important in the processing of the genetic material of the viruses. First, they may have DNA polymerase, and that is the full name of this polymerase. Then they usually produce some so-called scavenging enzymes that are converting RNA to DNA and phosphorylates the DNAs to provide substrates for replication. So these are desoxyribonuclease, ribonucleotide reductase, thymidine kinase, and a lot of proteases. So out of the variety of the enzymes, we have substrate-specific viral enzymes. What does it mean, substrate-specific? That's viral, typical with the particular specificities characteristic to the only those types of polymerases or reductases or whatever. So the very specific enzymes produced by DNA viruses are DNA polymerase, ribonucleotide reductase, and thymidine kinase. And why it is so important for us? These enzymes that play with the viral genomes, they differ from that of the cellular origin. They are different from the cellular analogs. So that's why if we affect the viral enzymes, we won't affect so much the human RNA, DNA converting processes, right? So that's why these three enzymes make the potential targets for antiviral chemotherapy. Few words about the structure. Inside the virion, we have a DNA core that is surrounded by icosahedral capsid. You may see here the icosahedral capsid made up of, as typically for cubical viruses, it is made up of 162 capsomeres, and they demonstrate typical hexagonal structure. This structure is called nucleocapsid. And the nucleocapsid, this is DNA together with the icosahedral capsid. Nucleocapsid is covered by the glycoprotein containing envelope, like any other enveloped viruses. And I would like to remind you that this envelope is partially cell-specific because the envelope, that is the membrane, derived from the previous host cell. But the difference with other enveloped viruses that herpesviruses multiply inside the human cell nucleus. So that's why mostly the envelope is taken out of as a part of the nuclear membrane, but not cell membrane. And then in between the envelope and the nucleocapsid, we have a space that is called tegument. And it is filled with the viral proteins that are very important. So these proteins have multiple functions in the processes of transcription and translation of viral genome. They may interfere with the processes of replication of the host cell. They're supposed to shut down the host cell replication, the host cell genome transcription and translation. Then may cause transactivation of the expression of the first set of genes, because the genome is very large. So the transcription of the genome cannot be made at once, right? So that's why it is usually performed step by step. And the virional proteins that are found in the tegument, those enzymes, are very important in initiating this process. So then on the surface of the envelope, we have surface glycoproteins that polymerize and make spikes, and here they are up to 8 nanometers long. And altogether, the Herpesvirus may have up to 11, and now we think up to 13 different glycoproteins. And they have multiple functions. First, they mediate the attachment of the virus to the cell surface receptor, and these are glycoproteins BCD. So then they may perform fusion function to fuse the viral envelope with the cell membrane, and these are glycoproteins B and H. Then several glycoproteins are responsible for evasion of the immune response, because they may reduce the antiviral effectiveness of the antibodies, and then they also may interfere with component activation. So because glycoprotein C may bind with the crucial C3 component of the complement system, so they may after that, this fraction can be depleted from the serum. So then the complement activation won't be possible. So then the Fc portion of the immunoglobulin G usually binds with glycoprotein E and I, proteins E and I. So, and that's appears something like in Staphylococcus and its protein A. When the immunoglobulins bind with not FAB portion but with FC portion, then it helps to camouflage the viruses and virus-infected cells, so they won't be recognized as foreign by the factors of acquired immunity. Herpesviruses have double-stranded linear DNA genome, and it is quite large. And then this genome encodes up to 100 different proteins. I would like to underline, my dears. 35 of those proteins can be found as structural in a virion. But the rest of these 100 proteins are virus-specific proteins and enzymes. They will be produced in a host cell while virus infected and undergo replication, but then those proteins and enzymes will be not enclosed into the ready-made virion. So they are non-structural proteins, the rest except for 35. Several virus-specific enzymes, like we mentioned already, DNA polymerase, thymidine kinase, they are synthesized in the infected cells, but they will not be incorporated into newly synthesized virions. So then we will discuss some general features of the Herpesvirus replication, because in each subfamily it may vary a little bit, so we don't pay attention at those variations. We will discuss the general scheme of viral replication. But before, how to divide this large family of herpesviruses? On the basis of the biological properties, pathogenesis of the disease, and disease presentations, these viruses can be divided into three subfamilies: alpha, beta, and gamma Herpesvirinae. So these are the official names of the genera and official name and common name, my dears. It's very good to know the numbers also, but also the common names of these viruses, because it may explain the usually the clinical presentation of the disease. So we have three types of alpha herpesvirina. This is the first subfamily. And here we have the most common herpesviruses. Number 1, 2, 3 human herpesviruses, and this is HSV1, HSV2, and the third one is varicella zoster virus. What are the common biological features of this virus? First, the duration of the growth cycle, whether it is short or long or variable. These viruses have short growth cycle. So it means they multiply very fast. And also usually they cause cytolytic infection. So when they release from the host cell during the intensive propagation of the virus, they cause lysis of the cell for several reasons, due to the damage of the nuclear membrane and cell membrane and other things. But then the cell's gonna die quite quickly. Then they are of same tissue affinity. Usually these viruses are called neurotropic because they used to cause latency in the sensory nerve ganglia. So, and the primary infection occurs usually in mucoepithelial cells as a primary target cells. And they spread usually through close contact or aerosol droplets or sexual contact. So very common is the direct contact except for sexually transmitted. So the second group is beta herpesviruses. So here we also have 5 different types. These are human herpesviruses 5, 6, and 7. Number 5 is called in another way, common name, Cytomegalovirus. And human herpesviruses 6 and 7 don't have the particular common names, but because we learn what do may cause in humans not so far ago, but then we found some diseases, at least one, roseola infantum, that may be caused by human herpesvirus type 6. Then later on we take a look at them. So, and we will discuss the biological properties of beta herpesvirina a bit later on. And the third group is gamma herpesvirina. So here we have 2 types of the viruses, lymphocryptovirus, that is Epstein-Barr virus number 4, and rhinovirus genus, Kaposi-sarcoma-associated virus, or human herpesvirus type 8. Now we start with the replication, and then we additionally discuss the biological properties of all those subfamilies. So the most typical virus is HSV1 and 2 viruses. So that's why the replication of the herpesviruses, the features are given for HSV1 and 2 here. Usually it takes for them around 18 hours to multiply, while the cytomegalovirus may multiply, for example, one cycle may take 70 hours. So cells productively infected with the alpha herpesvirus usually are killed because normal cellular DNA processing and protein synthesis will be stopped with the special protein that are incorporated into the tegument of the virion. So, and for different types of subfamilies, the cytopathic effects of these viruses can be different. Now first what we have, and then we discuss generally the replicative cycle. So first we know that the immune cells, the innate immunity cells supposed to recognize the microbes according to their pumps. We think that the viral glycoproteins, then genomic DNA and RNA structures that occur during replication may serve as the herpesvirus pumps. Then we mentioned that minimum of 11 glycoproteins are found on the surface of the envelope. Some of them are conserved, and these are glycoproteins HBL. Then they may help in entry, egress, host evasion, and cell-to-cell spread. So we repeat that in teguments we have proteins with the functions of host RNA degradation. It's supposed to be destroyed, not to be translated. Then viral genes expression, because it is very complex and supposed to be performed stage by stage. Then they also may help in host evasion of the immune response, and host cell activation. So a few words about the viral genome. It is quite large, up to 220 kilobases. Then they have very high GC context, 47 to 71, and this is different. And they have conserved adenine-thymine-rich regions such as ORES, originated of the transcription. Okay, and then we take a look at the replicative cycle. And to summarize it, we have to mention that we divide the cycle into substages because the virus is very complex, so that's why the process of replication is really complex. Okay, then we start with the attachment of the virus to cellular receptors, and those receptors are quite often ubiquitous, something like glycosaminoglycans of the cell membrane. So that's why they bind with it, and then they enter by fusion, mediated by the surface viral glycoproteins. And then the capsid that will be released together with some very special virional proteins will be released out of the envelope into the cytosol, and that is partial uncoating already. And then some very important proteins together with the viral nucleocapsid will be transported into the nucleus, my dears. That's important because those viral proteins supposed to help in the shut off the cell RNA translation and instead initiate transcription of the viral specific RNAs. So the replication occurs in several stages. We may develop out of up to 8 substages. In the first substage, as soon as the viral nucleic acid will be released into the nucleus and the capsid proteins will be left outside, that is a second stage of the uncoating. Then the viral genome will be fixed as a circular. And then the transcription supposed to occur. So the RNA should be produced out of the viral genome DNA. But then for this, uh, not the whole genome is transcribed at once, but the genome will be transcribed portion by portion. So then the first portion will be transcribed with the help of the virional proteins that are there. And then the resulting mRNA of the so-called alpha genes will be transported to the cytoplasm and on the ribosomes. Their translation of early genes into early proteins will be produced. Those immediate early proteins will be produced. Those alpha proteins will be then, take a look, taken into the nucleus. Why? Because their function is to upregulate the transcriptional and translation of the next portion of the viral genes. So after this, the early genes transcription occurs into RNA, and the second RNA here, it will be again transported to the cytoplasm, and second portion of the protein that are called beta proteins or early proteins will be produced, and then mostly transported back into the nucleus. What will be the very important function of these beta proteins? To perform or to help in transcription of the replication of the viral genome. See, the viral genome will be replicated already. So it means that the viral genome will be transcribed, and first the conchimeric DNA will be produced, and then it will be transcribed, and the late gamma genes will be exported into the cytosol, and the late proteins will be produced. And my dears, out of the late proteins, not all of them will be transported to the nucleus. Those that are cleaved and in red color, they will be transported into the nucleus to make the nucleocapsid for the newly synthesized DNA molecules. So they will be enclosed into the empty capsids. And then these empty capsids start to be released from the nucleus through the nuclear membrane. But my dear, those proteins that are not transported into the nucleus, they will be transported through the Golgi apparatus, and they will be inserted into the membranes of the nucleus and Golgi apparatus, and viral nucleocapsids start a series of budding from the nuclear membrane and endosomal or the membranes of the EPR. So due to the multiple series of budding, then finally the virions obtain all glycoproteins they need to keep, and then they will be released from the host cell by exocytosis or cell lysis, and then the newly synthesized viruses can be then participate in new cells infection. So that's how it occurs. And that is the overall. See, the capsid is capsid is delivered due to the entry of the virus. Then the viral DNA will be released into the nucleus. Then, step by step, immediate early genes will be transcribed and viral proteins produced. Then they will be taken back to the nucleus to regulate the early genes transcription and proteins translation. And then they mediate the DNA replication or DNA synthesis for new progeny virions. And then the late genes will be transcribed and late proteins will be translated. And those usually late proteins participate in this building up the structure of the virion with the capsid with the DNA inside, and this is capsid. Then through the cytosis from the Golgi network, they will get sufficient amount of and the set of glycoproteins, and then they will be released of exocytosis or cell lysis. So here you can see the release of Herpesviruses from EPR or vacuole that release their content at the surface of the cell, and then the virus will off of the cell. So now a few words about the diseases caused by herpesviruses, and of course we start with the alpha herpesvirus family, HSV1 and 2. Usually they infect epithelial cells and establish latent infection in neurons. So from this we think these are neurotropic. And here you can see the most typical herpesvirus. This is really one infection, and it is usually associated with the oropharyngeal lesions, and very commonly cause a recurrent attack, and that will be called the fever blisters. So HSV2 primarily infect genital mucosa, and the HSV1 we used to call a disease the herpes labialis, while the HSV2 usually called herpes genitalis, and it may progress sometimes with cervical or into the cervical or vulvar carcinoma. So that's why we think that HSV2 is also oncogenic. Both types of the viruses may cause neurological complications of the previous acute infection. This is sporadic encephalitis and severe neonatal infections, mostly caused by HSV2. But then if you take a look at the scheme of the sites of the body where herpesviruses may cause infections, you can see that quite often we may not see that much difference in the spreading of the diseases. So, but then HSV are more common. Usually, if the infection cause in clinic below the waist, we consider it as HSV2, and above the waist, we consider it as HSV1. So they are quite common, HSV1 and HSV2, but still some of their characteristics may be different or distinguished. For example, restriction enzyme analysis of viral DNA gives the different patterns. So then they may vary in unique proteins. For example, some of the glycoproteins may be specific for particular type, HSV1 or HSV2. So then we have a slightly different mode of transmission. Usually HSV1 is transmitted by contact with infected saliva, and more commonly HSV2 is spread by sexual contact. But that can be upside down. So differences in clinic features of human infection also occurs caused by HSV1 or HSV2 that we have discussed already. So what is the about pathogenesis of the disease? Usually primary infection may occur by HSV1 through the contact of through the mucosal surfaces. For example, broken skin may contact through kissing, for example, with infected excreta, or through the contact with conjunctiva, or in case of HSV2, that's most common genital herpes. This is the genital area will be the site of entry, or quite often both of them may enter through the vitro. Then they cause active multiplication, and replication of the virus occurs, and then the primary clinical symptoms will appear. But then, after the series of replication occurs, the virus will be depressed by the appearance of the antibodies in the blood, and then they will travel to from the primary site of entry of infection through the local nerve ganglion to the sacral nerve ganglion if we are talking about genital herpes, or most commonly the HSV1 may travel through the trigeminal nerve ganglion from the that innervates conjunctiva, nose, and the mouth. So then, quite often there the persistent infection occurs. This latent infection resides, virus within the latent infection resides in ganglia, and again the trigeminal nerve, quite often brachial plexus ganglia or sacral ganglia. They occur there in non-replicating state. So it means within this time, with the help of the typical assays, we cannot find the virus in the body, but antibodies will be there. So usually primary infection of the HSV1. Okay, and then, my dears, once again, but there, in those places, the viral will stay forever. So we cannot eliminate the virus from there. The primary infection usually appears in small children as the infection of buccal and gingival mucosa of the mouth with the gingival stomatitis, gingivitis, temperature, sore throat, vesicular and ulcerative lesions on the cheeks, on the border between red border in between lips and the epithelial cells, also may cause anorexia and malaise. So if the primary infection occurs in adults, it may appear as pharyngitis and tonsillitis. And then they travel to the trigeminal nerve, but then in trigeminal nerve, virus may stay for a very long while in non-replicated state. But if it is if it is activated by some stimuli like over-exercise in the gym, or emotional or physical stress, or over chilling, for example, or overexposure to sunlight, the virus can be so-called reactivated from the latent infection. Then the infection that will be the result of activation of the virus that stayed already in the body will be called reactivated herpes or recurrent infection. So we mentioned what provocative stimuli may cause this infection. And then the virus following axons go back to the peripheral sites and replicate again in the same cells where it causes usually primary set of replication. And it is important to understand that immunity in the host that is developed, and it will be the already immunoglobulin positive in individuals. But then this antibody cannot act locally and cannot prevent the recurrent infection of the HSV1, but also HSV2. The typical clinical presentation of the recurrent infection is called cold sores or fever blisters that occur around the lips. But it's very common when it happens in conjunctiva, because the initial infection may occur through the eyes, right? And then it is capable of serving very severe illness. So that is usually clinically presented as keratoconjunctivitis. And then the recurrent infection may cause keratitis or corneal ulceration or vesicles on the eyelid formation. Then progression of recurrent keratitis may cause blindness. So you can see here that that's how the initial corneal infection occurs. Then after multiplication, virus by retrograde transportation reach the trigeminal nerve and cause latency there. Then when it is reactivated, it travels back to the primary site of entry, and this is anti-retrograde transport through neurons, and renew corneal infection and multiply there again. That's why it may usually cause blindness in severe cases. So cutaneous infections, both from, for example, whitlow, may also be caused by HSV1 and HSV2, and that is usually called traumatic Herpes. That also may occur so-called Herpes gladiatorum when we have lesions on the bodies, and usually it occurs in boxers and wrestlers. Severe cutaneous infections may occur in patients with the eczema when the skin is permanently irritated, or in cases in burns. Then as a result of continuous HSV skin recurrent infections, the eczema herpeticum may be developed, and this eczema appear to be very difficult to be treated. So then, as a result of the transportation to the trigeminal nerve ganglia, so it is believed that especially HSV1 may cause sporadic fatal encephalitis as a result of direct infection in the trigeminal nerve. And the patients who survive this infection usually have residual neurological defects. But then it is believed that the HSV1 may reach the brains by several routes, from the ophthalmic branch if the virus infects through conjunctiva, then through the maxillary branch and mandibular branch from the nose and from the mouth infection. So they reach trigeminal nerve and multiply there. And when something goes wrong or immunity is depressed, so then directly the virus may travel forward and cause the lytic infection, very commonly in the temporal lobe of the brains. That is really very severe complication. So a few words about genital herpes. So it may be gotten mainly by sexual contact. And if we have the oral sexual contact, oral intercourse, so then they may occur in the places where HSV1 are more typical, in the oropharynx, for example. So herpes HSV2 is characterized by painful vesicular ulcerative lesions. Usually here you can see the formation of this in the penis, and in female it is formed in cervix, vulva, vagina, or perineum of the female. Usually it is accompanied with fever, malaise, dysuria, and inguinal lymphadenopathy, and then the genital and extragenital lesions will be performed. So then extragenital lesions may be complicated if it travels through the trigeminal nerve with meningitis. Recurrent infections are usually milder than primary infections and can be even asymptomatic. That's the traveling of the genital herpes during the infection. So first, establishment of latent infection, so HSV1 still. So that's also occur with HSV2 also. So the primary infection causes the multiplication of the virus, then the virus enters through the axon terminals, and by retrograde transportation it occurs into the trigeminal nerve, and it is believed that some viral protein 16 for example, and the herpes cellular nuclear protein also participate in the formation of the reactivated herpes. So usually the viral DNA occurs in the cells in a form of episome, my dears. Please, you can see that the virus usually, the viral genomes don't enter the human chromosome, and then it occurs as in a form of episome. Then if we have nuclear accumulation over the viral protein 16 and HCF1 supporting immediate early gene expression, then it may lead to the reactivation of the herpes virus, and then it travels back to the primary site of entry, multiply, and shedding of the new HSV variants occur. And then the reactivation herpes happens again. For HSV2, but also for HSV1, the newborn infections are typical. They may occur in utero, during birth, or postnatally. So then, neonatal herpes virus infections are very severe. So you see clinical manifestation of one of these, and they may cause several categories of the diseases. So they may cause formation of the lesions localized on the mouth, eye, and skin. Then they may progress to encephalitis or disseminated disease where the multiple organs are involved. All of those diseases may have clinical symptoms with very high mortality rate. And the cause of death of the babies is usually occur out of viral pneumonitis or intravascular coagulopathy. So how we perform lab diagnosis? Usually isolation of herpesviruses, of alpha herpesviruses, can be performed and not so difficult to perform in cell cultures like HeLa, human embryonic fibroblasts, or some other cells. So then they produce, because they multiply very quickly, they quickly produce cytopathic effect on the first third day of replication, and usually syncytial formation because they have the ability to fuse with the cellular membrane. So the syncytial formation or giant cell so-called formation is very typical. That is cytology can be used, right? Then the immunofluorescent staining may occur, may be used to perform the immunofluorescent assay, or neutralizing test can be performed in cell cultures. Then the restriction of endonucleases assay are used to demonstrate the difference between HSV1 and HSV2 mainly. My dear, serology is not of high diagnostic value. It may be used to confirm the primary infection in herpesvirus negative individuals. So it means they don't have antibodies yet. So, but then the serological tests are not really useful in the diagnosis of recurrent infections because the very high prevalence of the disease make the finding of antibodies not so interesting. So then, few words about the development of the treatment. So, the nucleotide analogs are inhibitors of the viral DNA polymerase, and that is the most common target for antiviral treatment of herpes virus infection. The first one was Cyclovir. This is the most prescribed anti-herpes virus drug by now, and relatively it is non-toxic. And then you can see that when Cyclovir is given, it's supposed to be activated by viral thymidine kinase, and that is very good because if the no herpes virus infection, the Cyclovir or the other one, Pathogenesis, that come from different types of nucleotide analogs, they won't be inactivated. They won't be activated. So, and once they activated, they form Cyclovir or pancyclovir monophosphates with the cellular kinase will be activated and transformed into acyclovir triphosphase or pancyclovir triphosphase. And that this drug are very effective in the preventing of the elongation of the HSV DNA. You can see here that this process will be stopped by these two types of anti-herpes virus drugs. So some other types like famciclovir, valacyclovir, vidarabine, They are less soluble, less potent, and more toxic than Cyclovir. By now, it is considered as the best drug against HSV1 and 2. How they can be prevented, my dears? No vaccine are currently available against HSV1 or 2. So the only good level of sanitary and hygiene and avoidance of the direct contact with an active mucocutaneous lesions may help us, my dears. It doesn't mean that the person infected latently with HSV1 or 2 is permanently infectious. No, my dears. The person is infectious only when the mucocutaneous lesions are seen on the body, because only then the person may shed the viruses into the environment. If it is in the latent stage, in non-replicable stage, the person is non-infectious, and this is true for both HSV1 and HSV2 infection. Few words about chickenpox and shingles virus, that is called varicella zoster. Two names of the virus, so two diseases. Varicella causes chickenpox, zoster causes the formation of so-called shingles. Usually, chickenpox occurs as the primary infection caused by varicella zoster virus. It is usually acute, but it is very common, highly contagious, but hopefully quite mild, and occurs usually in children. It is characterized by fever and the development of the generalized vesicular eruption of the skin. And the thing is that at the same time on the skin, we may see all stages of those lesions development, starting from macular, papular, vesicular, and pustules are formed all together, and of course at one time. And the mortality rate is very low, while in immunocompromised children, this virus may cause quite severe complications like encephalitis, varicella pneumonia, or disseminated disease with very severe sequelae. Then what happens when we have the initial infection? Virus multiply and travel to the neurons. And then when the latent infection is established in neurons, upon reactivation, the viruses from the dermal nerve may travel to the site of primary infections and cause there the disease that is called sporadic, and usually it occurs in adults, and it is very common in immunocompromised individuals where we have reactivation of this virus very commonly. My dears, you can see here the very clear clinical manifestation of the shingles that is accompanied with severe pain, and rash is usually limited in distribution to skin innerved by a single sensory ganglion. And usually eruption is usually unilateral, and the common complication of this neuralgia, and people who are infected for the first time with the varicella zoster virus are apt to develop serious fatal pneumonia. And we can see here the pictures of HIV positive individuals with very severe immunocompression. That's how the disease may appear, shingles may appear in HIV positive individuals. Usually, to confirm the disease, the laboratory diagnosis can be performed with the help of the histology detection of the giant cells, and it occurs in scrapings of swabs of vesicles. Then intracellular viral antigens can be demonstrated by immunofluorescence. And electron microscope is not very common. Of course, it can be used. Serology, when we perform the paired sera, then it can be used with the typical antigen in various tests like complement fixation test, neutralizing test in cell culture, and ELISA is very common. So for treatment, usually we don't use anti-herpesvirus drugs in children because usually varicella is a very mild disease. If the disease is very severe, it occurs in immunocompromised or very weak babies, immature babies, then the varicella zoster immunoglobulins can be used at the very early treatment. Then nucleotide analogs also can be effective. To prevent the varicella zoster infection, primary infection, a live attenuated varicella vaccine is been approved already worldwide. But then, if the disease occurred naturally, the vaccination cannot prevent the reactivation of the virus and shingles development. Okay, we finish with the first subgroup and we take a look at the second one. Out of there, we have Cytomegalovirus and human herpesviruses type 6 and 7. What biological properties are typical for that group? So, they are slow-growing viruses. And they grow inside the nucleus, and they may cause so-called cytomegalovirus when you compare with other nuclei. So, and they increase of the nucleus in size. And this is for CMV, and HHV6 and 7 may cause long lymphoproliferative disease because they are lymphotropic rather than neurotropic. So that's why we consider that the site of latency for all these viruses is secretory glands and kidney for CMV and lymphoid tissues in general for HHV6 and 7. And the primary target cells, epithelial cells, monocytes, and also T cells for HHV 6 and 7. The infection of the beta herpesvirus may occur through the close contact, blood transfusion, organ transplantation. It may occur congenitally or through respiratory route. That is more typical for HHV 6 and 7. What do we know about cytomegalovirus, or do you need to know? Of course, we know a bit more. They share the typical morphology with other viruses, and the name is derived from the cytopathic effect caused by these viruses that cause massive enlargement of the virus-infected cells. They replicate in epithelial cells, respiratory tract, kidney, and salivary glands. But then, for persistency, they enter lymphocytes and persist there for a very long while. CMV is widely dispersed throughout the world. And the very important property of CMV, that it is a very mild virus. And the infections are usually asymptomatic in immunocompetent population. And the prevalence of antibodies against this disease in adults is somewhere in between 40 and 100% depending upon the socio-economic and economical conditions. So, but then, why we bother so much about CMV virus? Oh, yes, as we said, in the adults it causes asymptomatic or very mild disease, but then it is really dangerous for congenital infections, because the congenital infection may result in death of the fetus. That is really important. So if we are talking about pathogenesis, the most infections are subclinical in both immunocompetent and immunocompromised individuals. Sometimes we may have symptoms of so-called febrile mononucleosis. So, but then the disease caused by CMV is generally called infectious mononucleosis-like syndrome. It is not typical infectious mononucleosis caused by another herpes virus that is called EBV virus. So, but then CMV causes the disease that a bit similar with that of the EBV virus, because it is maybe characterized by malaise, myalgia, liver discomfort, protracted fever, and lymphocytosis. Usually this is a very mild disease, and also subclinical hepatitis may occur. Complications are very rare in immunocompetent individuals. But then, if we come up to the immunocompromised individuals, CMV often develops more extensive organ disease with potential fatal consequences. The most typical clinical sign of the CMV infection in HIV positive individuals is interstitial pneumonia, what is the very common and connected with HIV positivity. So also it may occur in cancer patients and others. So except for this, it may cause retinitis in transplant patients, and even encephalitis, radiculopathies, and endocrine disturbance in HIV-infected individuals. That is the second. So first, it is asymptomatic or very mild in immunocompetent. Then it causes severe, very severe infections with interstitial lethal pneumonia quite often in HIV individuals. And then the third that we need to mention, that what they cause in fetus. And at birth, most 85 to 90 children are normal. But from 5 to 10% of infants who are infected with CMV may not develop symptoms at birth, may have varying degrees of hearing and microcephaly loss, mental or coordination problem, and even microcephaly. That is possible when the pregnant woman within the first trimester of the infection is infected with CMV. 10 to 15% of generalized infection may occur in the rest of the infected, and symptoms may range from moderate enlargement of the liver and spleen with jaundice to very severe fatal illness that we see here. How do we perform laboratory diagnosis? So it is performed usually first for those who are planning to get children. So then it is better to know whether mother and father are CMV positive or negative. It doesn't mean that they cannot get babies. But then it appeared that the CMV virus should be monitored where we have the latent stage of the disease or recurrency of the disease. It can be performed by ELISA to see the CMV viral antigens or immunofluorescent assay. So then visualization may occur with the electron microscopy or DNA-based molecular assay. Usually cell culture method is based on isolation of the virus from washings or throat in cultures with the appearance of cytological changes that are the owl's eyes inclusion bodies formation. See, like bird's eyes. Serology can be performed by detection of CMV specific antibodies by neutralizing immunoassay or immunofluorescent assay. So usually if we have the CMV infection immunocompetent individuals, we don't perform any treatment. But if we have life-threatening cytomegalovirus infection in immunocompressed individuals, like we mentioned, very severe retinitis, interstitial pneumonia, colitis or esophagitis, so then some drugs can be used. Usually these are again Cyclovir and Foscarnet. They are recommended because they can be used in lesser concentrations to compare with acyclovir or valacyclovir. Some other preparations like recombinant interferon alpha or interferon inductors can be recommended. How to control? We don't perform vaccination. It is not available. So then isolation of newborns with generalized cytomegalovirus infection and screening of transplant donors and recipients for cytomegalovirus infection may prevent CMV transmission within organ transplantation, blood transfusion, and during the pregnancy. Cytomegalovirus immunoglobulin may be administered to decrease the risk of this infection in the transplant patients. There are some experimental vaccine candidates, but by now they don't give any good protection against this disease. Few words about HHV6. It was discovered initially in 1986, not so far ago, from AIDS patients with lymphoproliferative disorder. They have greater than CMV tropism for T lymphocytes. In 1998, it was discovered that HHV6 was the causative agent of ubiquitous childhood disease that is called exanthema subitum, commonly referred to as roseola. And interestingly, that seroprevalence of HHV6 infection is estimated to range from 90 to 95% in adult population of the world. Maybe this is the most common herpes virus infection. One more very interesting thing, we stated that human herpes viruses occur in the infected cell nucleus in a form of episome not integrated with the human chromosome. But for HHV6, it was shown the chromosomal integration in approximately 2% of seropositive individuals and is often the cause of high level of viral DNA in host serum and plasma. And you can see here with the fluorescent assay visualization of HHV6 in the integration into the telomere region of the chromosomes and 9 in metaphase and interphase cells. See, this is the integration of the virus into the chromosomes here, right? HHV6 is required usually in early infancy and causes roseola infantum or exanthem subtilis. It is characterized by very high temperature, usually before it was underdiagnosed febrile illness, and it occurs usually for 3 to 7 days. After cessation of the fever, approximately 17 of infected infants develop this roseola. But then the rash persists in 1 to 2 days, and it is not increase the transmission of the virus, because the rash is mainly to the because of the immune response, and the new virions are not really shedded with the rash. So then the rash occurs like this, and usually doesn't cause any itching or troubles to the infected individuals. So additionally, it may cause fatigue, gastrointestinal and respiratory symptoms, but then the most dangerous is febrile seizure that may occur during the acute initial stage of this disease. High levels of viral titer have been isolated from the brains and cerebrospinal fluid of those who are infected, and can be associated with rare CNS symptoms such as encephalitis of unidentified nature. So then usually HHV6 after the initial infection cause latency, and usually persist in healthy seropositive adults and children. And that is the second stage of the infection, latency, after the acute infection. Viral replication continues during the latency in the epithelial cells of the salivary glands during this stage, right? And it accounts for the very broad spreading of the virus and shedding of the virus with the saliva samples, and that is a primary means of transmission to seronegative individuals. Maybe that's why the prevalence of this virus is so high, I would like to remind you, from 90 to 95% in the population. Then the most acute clinical manifestation of HHV6 infection is observed during the reactivation of the virus in immunocompromised individuals, mainly organ transplant patients and AIDS patients. It is believed that HHV6 may increase the HIV replication, but upregulating certain cytokines and transactivating of the LTR fragments of the HIV genome. We will take a look at them next week. So, and then it increases the viral load, and in immunocompromised individuals infected with HHV6, the disease, the HIV goes to be more aggressive. And here you can see the sketches, how to recognize these three very common children exanthemas. That is ordinary measles, or that's called rubeola. Then German measles, that is caused by rubella virus, and radiola infantum, that is exanthema subitum. So you may read and compare these symptoms. That will be good for children disease. So then human herpesvirus 6 infect T lymphocytes. It may grow in cell cultures of B lymphocytes also. And they are required in early infancy and cause roseola infantum. Then that is the clinical manifestation of the disease. It is believed that HHV7 infection may cause so-called chronic fatigue syndrome. That's typical for students, isn't it? So now a few words about gamma herpesviruses. So we have two Epstein-Barr virus, HHV4, and Kaposi sarcoma virus, that is HHV8. So their biological properties. The primary target cells are B lymphocytes and epithelial cells for EBV, and maybe the same for HHV8. But then the site of latency occurs in lymphoid tissues, and the primary target for EBV viruses are B cells of almost all stages of their differentiation. So, and EBV is called kissing disease because the most common transmission occurs in children and teenagers through kissing. But EBV replicates in epithelial cells initially due to the acquisition of the oropharynx and parotid glands. But then it reaches B lymphocytes and establishes latent infection there. And it also causes acute infection there, and the disease caused by EBV is called infectious mononucleosis. And these three complications are due to the infection sequela that occur in immunocompromised individuals, especially in AIDS patients, and they are associated with diffuse polyclonal B cell activation and formation of lymphomas, lymphocytic interstitial pneumonia, hairy oral leukoplakia of the tongue. So, how EBV occurs in healthy carriers? Primary EBV infection begins in oral cavity usually, and EBV propagates in the epithelial cells first. And then it will be transported to the B cells. They infect B cells because they have a special receptor, CD21, that occurs on the B cells only. So through this receptor, they infect B lymphocytes and transport the viral genome into the B cell nucleus where replication begins, and EBV gene products activate the B cell growth program, resulting in the proliferation of blasting B cells. And then as a result of the proliferation, priming of naive T cells will occur against the Epstein-Barr antigens in parallel with the dendritic cells. Normally, those infected B cells are destroyed by T cytotoxic lymphocytes because it is believed that the EBV infection is the so-called civil war of T cells against infected with EBV B cells. So then the lysis of the infected cells with the help of TCL occurs. But once in circulation, previously activated B cells may continue to undergo lytic replication, or if EBV shuts down the most of its protein-encoded genes, latency may occur. In a latent time, as cells circulate between the oral and peripheral compartments, resting B cells may be activated again, and it may result in virus activating and shedding. So this circle occurs again, and again. So within the life, the many cycles of B cells reactivation and proliferation and virus shedding may occur. So pathogenesis. When the virus attaches to infected epithelium of the throat and infect epithelium of the throat, it replicates, causes pharyngitis, and then through lymphatic vessels it goes to the lymph nodes. So the infection may be localized in the lymph nodes. Then virions escape from the lymph nodes and go to the bloodstream where they multiply and specifically infect lymphocytes and infect them and produce them latency or productive infection. And then the latently infected B cells may be activated and then replicate and differentiate and produce random immunoglobulins. So because the polyclonal non-specific activation of T lymphocytes occurs. So not antigen specific. Do you understand what does it mean polyclonal non-specific? Without the recognition of the antigen. And then this causes the many polyclonal plasma cells activation. See, they are infected with EBV. So they start producing antibodies that are called heterophil antibodies that are not against particular antigen that had been in a body once, but then against some very strange antigens. For example, from the sheep erythrocyte antigens. And this simple test may help to reveal the so-called heterophil antibodies development. When we take blood and from the suspected EBV infection and combine the serum of this blood with sheep erythrocytes, if agglutination happens, it means there are heterophil antibodies there. So most commonly, these tell us that this is Epstein-Barr infection. So latently infected B lymphocytes usually become immortal and they may not be attacked by B cells. So then lymphocytosis may cause oncogenic transformation of the B cells and the development of the lymphomas. So I see that it is a bit over time. So the immunosuppression in immunosuppressed individuals, the EBV disease is usually very dangerous with serious fatal consequences like multiple lymphomas development because it is connected with the oncogenic transformation of the cells. So why the disease is called mononucleosis? Lymphocytes usually represent greater than 50% of blood cells, and at least 10% of them are reactive lymphocytes with granules. So that's why they are called atypical. So this is the mononucleosis that occurs during the EBV infection. So we have the very simple and sensitive EBV testing where for heterophil antibodies, and this is usually make the first stage of EBV diagnosis. So in the first stage we have a screening test, and that is rapid diagnostic test that is called monosport to find the heterophil antibodies against sheep erythrocytes. And then if it happens, these monosport antibodies are found. So then we perform more specific diagnosis by finding the specific EBV antigens. So this is lymphomas, my dears. Love comes and goes, but the herpes lasts forever. Any or out of 8 herpes viruses known by now. Goodbye, my dears.